everyone. Once again, I welcome all the students in our today's session. And as we have already started the chapter number 6, that is, the name is tissue. So, in our last session, we have seen or we have discussed so many points regarding this chapter. We have compared, we have started with our previous chapter, that is cell, and how it is related, means what is mean by the cell. It's a fundamental and basic structural unit of the life. And then we have seen the definition of tissue. So we have seen a number of or the same kind of the cells, whether it is same in functioning or in the structure, they can be perform a same function. So they are come together and they will perform a specific function, or we can say the cluster of same type of the cell, same in the sense of structure or in the sense of functioning. So it is called tissue. And then we have seen what is the difference between plant cell and animal cell, or why there is a requirement or why there is a change in between the plant cell and animal cell. So these are some points we have discussed in our previous lecture, that the availability of the food or the requirement of the food is more in the animal tissues, because animals are moving beings. They are moving from one place to another place, right, in the search of the food and shelters and mates. So, and as the plants, they are just standing in one place only. They don't require so much energy. So that is why most of the cells, they having a dead cells. They are just supportive cells, right? So the tissue also, so many tissues, they having the dead tissues only for the protection or for support. But as we compare to the animals are moving. We have to move from one place to another place. We have so many special kind of the functioning in our body. So many different organs and tissues are there. So that is why so many different kind of the tissues are present in animal as compared to plant. And then we have started this we have seen this activity, first of all, the very first type of tissue in plant, that is called Marismatic tissue, right? First of all, we have started with the plant tissues. So we have discussed enough about the Marismatic tissue. And what is a Marismatic tissue? It is a dividing cell. It is a type of the tissue which is having the cell which is having the ability to divide very fast, very rapid dividing abilities are there in the cells of Marismatic tissues. And it is present namely onto the tip of the roots or shoots of the plant, as we have seen this activity of the onion bulb in the previous lecture, how while it is cut from a tip of roots, in jar B, the roots is not growing so quick as compared to the jar 1. So this is with the help of activities we have seen the importance of the Marismatic tissue. And then we have seen this figure. So on the basis of the location, Marismatic tissue having a different types. So as we have seen this picture, that is apical meristem, because it is present onto the top of the plant, so it is called apical meristem. Lateral or intercalary meristem, because it is present near the node of the new branches. This is the stem, and from the stem the node is just starting. So there is also need of more growth. So whenever or where is the location where plants needs more growth, so in that region it is the Marismatic tissues are there, which is also called the dividing tissue. It is having the more ability to divide. And in the last we have seen the lateral meristem. So what is lateral meristem? It is present in the both the side of the stem, because it is providing the girth, it is providing the broadness of the tree. So that is why it is called the lateral meristem, on the basis of the location we have seen. So apical meristem is present at the growing tip of the stem, as we have seen, stem, root, and it is increasing the length of the stem and root. How we have seen the plant is growing roots for the search of water, and it is growing the shoots when it want to grow. It is growing towards the sunlight. So that are both the possible, because the apical meristem tissues are present into the stem and the root. That is why it is shown a more rapid growth as compared to the other plant. Or the girth of the stem or the root increases due to the lateral meristem. As we have seen, this is the lateral meristem. So it is responsible for the girth of the plant. So plant is just having the broad stem or broad roots, so due to the lateral meristem. It is also called cambium. So, and the last one, interclary meristem, is the meristem at the base of the leaves and internodes, where the new branches are starting. So where on either side of the nodes on twigs, so where the new leaves are growing, new branches are growing, that is called internodes. So that is why interclary, that is present into the nodes of the stem of the plant. So these are on the basis of the location of meristem. Basically they are having a 3 type. On the next page we will start here. That is, as we have seen, a different, the cells of these tissues are very active. That is why we have said that it is a very dividing tissue. They are dividing cells they are having. So they have a dense cytoplasm. Why? There is a reason that the tissues are very rapidly growing, or the cells are very rapidly growing. The reason is it is having the dense cytoplasm. So because of the dense cytoplasm and thin cellulose walls and prominent nuclei, they are having this ability to divide very fast. The nucleus is very prominent and they lack vacuoles, right? They don't have the vacuoles because it is only the dividing tissues. It needs only to grow and grow. So there is no need to store the foods. That's why they lack the vacuoles. And can we think would lack vacuoles? Why there is no need of the vacuole? This is the reason, because it is a dividing tissues, most rapidly dividing. There is no need to store the food. So that is why lack of vacuoles are there. So this is all about marismatic tissue we have seen. Now we will move through the second type. First type we have seen marismatic tissue, that is present on the areas of the plant which is growing very fastly, very rapidly. Now we will start permanent tissue. As the name itself is telling, it's a permanent, means already it has been grown and now it is on the stage of permanent, right? It doesn't require any more growth. So what happened to the cells formed by the marismatic tissue? While the cells we have started the growing in the areas of the tip of the plant, so that is a marismatic tissue as we have seen. So afterwards what will happen to the marismatic tissue? This is or they take up the specific role and lose the ability to divide. Once they are having enough growth, so the same tissues are just converted into the permanent tissue. The marismatic tissue only, they have the they lose the ability to divide and they will just convert into a permanent tissue. So we can also say permanent tissue is made from a marismatic tissue only. After enough growth of the plant, that same marismatic tissue will convert into a permanent tissue. So this process of taking up permanent shape, size and functioning is called differentiation. Now as we have seen, the marismatic tissue only, it is taking the permanent shape, size and functioning. So the process is called differentiation. So differentiation means the converting of marismatic tissue into permanent tissue. It is called differentiation. It is now taking a permanent shape, right? And cells of marismatic tissues differentiate to form a different type of permanent tissue. So as it is starting from a marismatic tissue only, then after the proper growth, it will just convert into a different type of the tissues that is come under the permanent tissue. So here there is a figure which is we will discuss later. It is showing the both the figures and section of a stem. This is actually a stem. Whilst we cut the stem and we are taking a small, very fine, we can say the section of the stem. So it is showing this kind of the cells are there. So in short we will just discuss about this figure. So here it is mentioning different kind of the cells. You can find a different not only the single type of the cell, that is what is called the permanent tissue. It is come under the permanent tissue. So as you can see here, this is the epidermis, which is the outermost layer of the plants, right? So here we can see this is the epidermis, means outermost layer of the plant. Then after that it is come the hypodermis. This is all both are the upper layer of the plant and cortex. Then under that endodermis is there and pericycle is there, phloem is there. Phloem, you can see here, this is the phloem. This part is actually shown the phloem. So what is the function of phloem? This is responsible to transport the food material. So as you can see, this phloem, which is present into the outside areas of the bark of the tree. This is actually the stem of a section of a stem. So in the stem the outward or outside layer of the cells after the epidermis, that is comes under the phloem, and phloem is responsible for movement of the food. And here is the xylem, you will see here. So xylem is present afterwards the phloem, that is internal layer of the stem. So now the xylem is responsible. The xylem is having tubular structures. We will see into the next slide. So it is having a tubular structure, and this tubular structure have the movement of the water from the roots to the tip of the plant. So that is it consists of different, you can see the different type of the cells are there. Here is showing the epidermis cells are also there, hypodermis cells, which is providing the protection to the plant, outermost layer of the plant. Then it is also showing the phloem, xylem. So the result of this slide, we can be see that it is consist of different type of the cells are there into the stem of the plant, right? So now we will just how we can get this kind of the slide. So this very clear, are we having this activity? So how we can be prepared like this slide? Actually this is the section of the plant which is see by the microscopic. This is the microscopic view of the plant, right, of the stem. We can, if you cut the stem of the plant and you can see into the microscope, so you will look like this. So for that this activity make you clear how you can do is. So for that you have to take a plant stem. With the help of your teacher you can be perform into this laboratory, and now take a very small cut into the very thin slice. Slice means very fine cut from the stem, and now just put the stem onto this slices. Stain actually required, actually here we are using the stain called safranin. Safranin is the name of stain, and what is mean by stain, which is need to be colored. If you are not using the stain and we are just want to look this into the microscope, so it will not look very visible. So that is why we are using a stain, and here the type of stain is called safranin. There is so many kind of the stains are there. Methylene blue is also we are using in when we are want to see the cells of the human beings. So safranin is which is comfortable for this slide. So we are just using the stain and place neatly cut section into the slide, and then put a drop of glycerin. So glycerin will use here, it will make the visibility very clear while you are looking into the microscope. Then cover with the cover slip and observe under the microscope. Cover slip, as you have performed before the practicals, it is covering to the material, it is providing the safety to the material, which is afterwards will not disturb. And then we will just observe into the microscope. So after looking to the microscope we will having this kind of the view, and here so many different kind of the cells we can be see here. So after observing to the microscope you will see the different type of the cells are there, and the arrangement is there, and we can be compare our slide with this diagram. So you will find this kind of the structures once you can be see into the microscope, like so. This is the section of a stem, and now we are in the stage of we can be give the answer. So all cells are the similar in the structure? So no, there are different structures are the cells we have seen into the slide. Some are xylem, some are phloem. And how many type of the cells we can be see? So many type of the cells we have seen, like for the epidermis, and then we have seen the xylem and phloem. So many kind of the cells are there which is present over there, and we can think the reason why would we have the many type of the cell. So why we have the many type of the cell? Because a plant have to be perform a different functioning. For that they have the different kind of the cell. They have the marismatic tissue, they have the again xylem and phloem and epidermis. So each and every cell having the different function. So as per the functioning we having the different kind of the cell, right? So up to here we have just seen the slide of a bark of a tree. Then we will start the simple permanent tissue. Now we are discussing about the permanent tissue. First we have seen marismatic tissue, that is most dividing type of the tissue. Now we have started a permanent tissue. So permanent tissue also having two types. One is simple permanent tissue, as we have seen here, that is the first type, simple permanent, and the other one we will see a complex permanent. So what is the difference between simple permanent and complex permanent? Simple permanent tissue, they are made from a same type of the cell. All the tissues, all the cells are which is making to this kind of the tissues, they are same type and same origin. That is what is called simple permanent. And complex permanent, the tissues which is made from a different kind of the cell, more than one type of the cells are there to make a tissue. That is why it is called complex permanent. We will see detail in next slide. So first we'll start with simple permanent. So simple permanent, a few layers of the cells form the basic packing tissues. So this tissues is parenchyma, as the packing tissue means the uppermost layer, we can also say, which is providing the protection. So simple permanent tissue also having a three type. Number one, we will start with parenchyma. Number two, collenchyma, and then we'll see the Scralanthyne. These are the three type of the simple permanent. So now we are just talking about a very first type, that is called parenchyma. So here the parenchyma is a type of the tissue which is consist of relatively unspecialized cells. They are the simple cells within the cell wall. They are having the thin cell wall, and they are leaf, or they are the alive cells. They are having the living cells. Like we have seen in a bark of a tree, so many cells are dead because it is only used for protection. But this section or this parenchyma, so they have, or they are leaf cells or the live cells, and they are usually loosely packed. When we are looking the arrangement of these cells in the parenchyma, so they are loosely arranged, means the cells are not compactly packed to each other. So once we are looking, it is a loosely arranged, so they are having the more intracellular space are there. So the large spaces between the intracellular spaces are more. What is intracellular space? The space between two cells when they are joining together and they are making the tissue. So as we have seen this kind of the tissues, they are loosely packed together. So if they are loosely packed, there is more intracellular space, and they are found in this tissues, and these tissues provide the support to the plant and also store the food. So what is the function of this parenchyma? That is providing the support also, and that is storing the food also. And in some situation it contains a chlorophyll, so it can be perform the photosynthesis, right? So this is actually the complex permanent, but sometimes this kind of the tissues, they are having chlorophyll, so that is why the name will be changed into the Chlorine. So that is a Chlorine, the type of the parenchyma which is having the chlorophyll pigments, and once it is having the chlorophyll pigments, it can be do the photosynthesis, and that is why it is called a Chlorine, because the presence of chlorophyll pigments are there. And it is, so this is the type of the Chlorine. And then if the parenchyma in the aquatic plant, when we are looking the parenchyma, once we have seen the parenchyma which is having the chlorophyll, so it is given the name Chlorine. So if the parenchyma cells are present into the aquatic plants, so they are having the large air cavities are present in the parenchyma to give the buoyancy to the plant to help them float. So when we are just talking about the aquatic plant, so into the aquatic plant, the parenchyma cells, they are having the more intracellular spaces, so they are having the vacuum inside, or cavity, air cavities are there. So this with the help of this air cavity, they can be float onto the water. They are having the very lightweight, or they can be having the buoyancy or a parenchyma. So that is why the plant can be float onto the water. So this is especially into the aquatic plant we will see. So because of that more air cavities are there, so it is the given name Aerenchyma. So this is Aerenchyma, is a type of same parenchyma, but if it is the plant is present into the water, aquatic plant, so it is having the more air cavities, so it is the given name Aerenchyma. Right? So the parenchyma of the stem and root also store the nutrients and water. So if the parenchyma is present into the roots, so it will also help to the store of the food and water. So especially the function of this parenchyma cells are there, which is providing the strength and providing the storing the food. So this we have seen very first type of the simple permanent tissue, that is parenchyma. Once we compare this parenchyma with this diagram, so this is actually the diagram of the parenchyma. So as we have seen, the cells of the parenchyma, they are loosely packed, so that is why the intracellular spaces are more. Here you can see, so it is showing here intracellular space, so it is having the more intracellular spaces. The space between the two cells are more, and if it is aerenchyma into the plant cell, it is also providing the means the plant can be float, it is having the more air spaces. So this is the structure of the parenchyma. And once we just see the another slide, this is showing the two slide actually. So in the other section, cross section, you can be see in the parenchyma cell there is a cytoplasm, there is a nucleus also present, middle lamina, and chloroplast. As we have seen, if the chloroplast is present, that is in the case of chlorine kinema, and vacuoles are also there, and intracellular spaces are more, as it is shown here, the intracellular spaces. So because of more intracellular spaces, it can have the floating ability. So this is the structure of this parenchyma. Then we will start the second type, that is called a chlorine kinema. This is going to be a second type of simple permanent tissue. So what is chlorine kinema? The flexibility of the plant. As we see, the plants are very flexible, it cannot be break down very quickly. So this is provided with the help of the another permanent tissue, that is a chlorine kinema. So here the chlorine kinema is a second permanent type or simple permanent type tissue. It is called chlorine kinema, and it is providing the strength or flexibility to the plant, and it allow the easy bending to the various part of the plant, where the plant can easily bend with the stand with a very blowing air. So it can be possible with the chlorine kinema only, and it is present in the leaves, into the stem, or without breaking. Once it is having some kind of the pressure, so without breaking it can be flexible, and it is an advantage. So it is also provide the mechanical support to the plants. So this is the advantage of chlorine kinema. And we can also find this kind of the tissues. This is actually the diagram of this chlorine kinema, second type. Here is the diagram of the chlorine kinema. So here the intracellular spaces are less, but the cell wall are very thick. Okay, so this kind of the tissues also present into the leaf stalk below the epidermis, right, below the epidermis, where the leaves are attached to the branches. So it is also present into the stalk of the plant, and the cells of these tissues are leaving. Most of the tissues of these cells are leaving cells, and elongated and irregularly thickened at the corners. So if you see the slide, the diagram of this chlorine kinema, so here the wall are very thick. So this section, this red color is showing the thick cell walls. So because of the thick cell walls, it is having the ability or flexibilities are there, and vacuoles are there, cell walls is very thick. So it is irregularly thickened at the corners. Onto the corners only the walls are very thick, and this is very little intracellular spaces. So when we are just talking about the intracellular spaces, so it is very less into this type of the cell, but the thickness of the cell wall is very high or dense. So this is also one of the view of that. Here we can see the thickness is more, and walls are written here. So this is the primary cell wall. Chloroplast is also there, nucleus is also there, cytoplasm, and intracellular spaces. So here it is showing the intracellular spaces are very less, because the cell walls are very dense or very thick. So it is also providing the movement of the leaves, or the leaves can be bent easily. And here the third type, that is a longitudinal section of the Scralanthyne kinema. So number third type, it is called a Scralanthyne kinema. So this is actually the microscopic view of the third type, Scralanthyne kinema. So here the cell walls are very dense, and because of the cell walls are very dense, the intracellular spaces are very or inside the cell space is very low or very thin. That is why it is look like a very narrow cells are there. So that is going to be a third type of simple permanent tissue, that is called Scralanthyne kinema. So both the figures which is showing here, that is the figures of Scralanthyne kinema. It is very narrow lumen, and it is having the very dense cell walls are there. So we will see some points about that. The third type of that, it is called a Scralanthyne kinema. So what is mentioned here, yet another type of the permanent tissue is Scralanthyne kinema. So this is going to be the third type of permanent tissue, right? And what is mentioned into this Scralanthyne kinema, it is the tissue which makes the plant hard and stiff, right? So when we see some part of the plant they are very hard, they are very stiff. So what is the reason? So in that area the Scralanthyne tissues are present, which can be provide the hardness to the plant or the part of the plant. And we have seen the husk of the coconut. So as everybody know, husk of the coconut are very hard and strong. It is providing the protection, and it is made up of the Scralanthyne tissue, that is made by the Scralanthyne, and the cells of these tissues are dead. So mostly the cells of these tissues are dead tissues. This is also just keep in mind. It is providing the strength to the plant, or the coconut husk is very hard, so the tissues are dead, and they long and narrow as walls are very thick. So the cell walls are very thick, that is why, because of the thick cell wall, it is very hard and thickened due to the lining. So what is the meaning of this lining? It is a chemical substance which acts as a cement, as we are using the cement for hardness in our building. So this lining is a chemical which is work like a cement, and it is providing the hardness to this part of the plant. And often these walls are so thick, so there is no internal spaces inside the cell wall. If the cell walls are very thick, so the intercellular spaces are very low, and that is why it is a very hardness, providing the very hardness. And this tissue is present in the stem around the vascular bundles to providing the protection. As we have seen, the stem of the plant, they are very hard and very strong, so that is because of this Scralanthyne tissues are present. So this tissue is present in the stem around the vascular bundles, veins of the leaves, and in the hard covering of the seeds and nuts. So if you see this, all the part, the veins of the tissue of the leaves and the hard covering of the seeds and nuts, so this is providing a more strength. It is a very hard part, so providing the hardness to this part, like nuts and seeds and husk of a coconut. So it is having this kind of the Scralanthyne. So this is the third type of permanent tissue we have seen, that is called a simple permanent tissue. So it provides the strength to the different part of the plant. So here we will stop in this lecture, and then we will the next part will start in our next lecture. So up to here we have seen a type of simple permanent tissue, right? So all the three types we have seen in our simple permanent tissue. So just go through this slide one more time, just see whatever the point we have covered, and these are especially the three type of simple permanent tissues. So that is going to be a parenchyma, scralanthyma, and cholenchyma. So this is the three type of the simple permanent tissue. In the next slide we will just start with the complex permanent tissue. So these are we are looking into the type of permanent tissues. First we have seen marismatic tissue, that is present in the dividing part of the plant, which is the plant dividing more rapidly. Then we have seen a complex type of the tissue. In that complex we have seen a simple complex tissues. Now we will, in the next lecture, will start with complex type of the tissue. So up to that, just rewind one more time this slide and write down whatever the important points are there, and see you in the next slide or in the next session. Thank you.